It feels like the search for an HIV cure has been going on forever.
It has been a long road for sure.
Yeah, but there's some new stuff that came out at the big HIV conference.
Yeah.
CROI 2025. It was in San Francisco
Yeah.
just a couple weeks ago, and it's got people talking.
Right. It's pretty exciting.
Yeah. So we're going to do a deep dive on this today.
Sounds good.
We've got these reports from Aidsmap that lay it all out, and the big news is that 2 more people seem to be functionally cured of HIV.
Wow.
Yeah, and this would make 8 and 9 now total.
Yeah.
Using this one procedure, it's stem cell transplantation.
Right.
And it's for people who have cancer.
Yeah. When they're getting a stem cell transplant for cancer, they also, try to cure their HIV at the same time.
Right, exactly. So
Yeah.
we're going to really try to break down these 2 new cases for you, like what actually happened,
Yeah.
what does it all mean for the future of a cure, and also, like, why can't we just do this for everyone right now?
Right.
So that's what we're going to dig into today.
That's a lot to unpack.
It is, but we're going to get those nuggets,
Exactly.
important stuff for you guys. So to set the stage here, can you kind of recap the cases that came before these new ones?
Yeah, so before these 2 new cases, there were a handful of other people who were reported as functionally cured of HIV after getting stem cell transplants for cancer. And, it all started with Timothy Ray Brown,
Oh, yeah.
the Berlin patient.
Oh, gee.
Exactly. So back in 2008, he got 2 stem cell transplants to treat his leukemia. And in both of those transplants, the donor had this special genetic thing called the CCR5 delta 32 mutation. And what this mutation does is it basically blocks HIV from getting into immune cells because it messes with this receptor called CCR5, which is the main way HIV gets in.
So it's like a locked door.
Exactly, a locked door. And so after the transplant, he stopped taking his HIV meds and the virus never came back.
Wow.
So he was HIV free for over a decade.
Amazing. And that was really like the proof of concept, right?
Exactly. It was huge.
It could be possible.
Yeah. And then after Timothy Ray Brown, there were a few other cases, the London patient, the Düsseldorf patient, and the City of Hope patient.
I've heard of those.
Yeah. And they all got stem cell transplants from donors who also had this double CCR5 delta 32 mutation. And none of them have had the virus come back either.
Okay. So it really seemed like that was the key, huh?
It really did seem that way.
The CCR5.
Yeah.
But then it's gotten more complicated, right?
Yeah, it definitely has. There's been some cases that have kind of thrown a wrench in that simple explanation.
Yeah, like what?
Well, there's the New York patient who got a transplant with a mix of cord blood cells that had the CCR5 mutation and adult stem cells that didn't. And then there's the Geneva patient who seems to be cured, even though the stem cells he got didn't have the CCR5 mutation at all.
Wild type, they call it.
Right, wild type. And then there's also the next Berlin patient who only has 1 copy of the CCR5 mutation, and his donor only had 1 copy too.
Yeah.
And he's in remission, so it's looking more and more like there's more to the story than just CCR5.
Right. It's a puzzle with a lot of pieces.
Exactly.
Okay, so let's get into these 2 new cases, starting with the Chicago patient.
Okay, so the Chicago patient is a 67-year-old man who had HIV for 14 years before he was diagnosed with acute myeloid leukemia.
Okay, so pretty long time with HIV before this.
Yeah.
So obviously he needed treatment for the cancer, right?
Right. So he got this reduced intensity conditioning therapy, which is basically chemo and radiation to get his body ready for the transplant. And then he got a stem cell transplant from an unrelated donor who did have the double CCR5 delta 32 mutation.
Okay, so that part's familiar.
Yeah.
So then what happened?
Well, about 15 months after the transplant, they couldn't find any HIV in his blood at all.
No way.
Yeah, his viral load was undetectable, and they couldn't even find any HIV RNA or DNA in his blood cells. And his HIV antibodies had gone way down, too.
Wow.
So they decided to stop his HIV meds.
Fingers crossed, right?
Exactly. But then, after about 2 months, the virus came back.
Oh, no.
Yeah, his viral load went up to almost 800 copies, so he had to start taking HIV meds again.
Oh, it's got to be so disappointing.
Yeah. It must have been a real setback.
But wait, there's more to the story, right?
There is. Even though the virus came back, they found something really interesting.
What's that?
So they couldn't find any HIV RNA or DNA in the new blood cells that he got from the donor.
Oh, so those cells were protected?
Yeah, exactly. And his T-cell responses and antibody levels didn't go up either, which means that his new immune system, the one from the donor, wasn't being attacked by the virus.
So the virus that came back must have been hiding somewhere else.
Right. It probably came from what we call the HIV reservoir, which is basically a bunch of cells that are infected with HIV, but just hanging out quietly in the body.
So it's like a sleeper cell?
Exactly.
Okay, so what happened next?
Well, he stayed on his HIV meds for almost 2 years, and then he decided to stop taking them again.
Brave man.
Yeah, and guess what? He's
Partner.
been in remission ever since.
Get out of here.
I'm serious. His viral load has stayed undetectable for 10 months now without any meds.
That's incredible.
Yeah, it's the first case ever where someone had a viral rebound after a transplant, but then went back into remission and stayed there.
So this shows that even if the virus comes back, it doesn't mean you can't still be cured.
That's right.
Wow, that's a huge takeaway for everyone listening.
Definitely.
All right, so let's talk about the second case, the Oslo patient.
Okay, so the Oslo patient was a 58-year-old man who had HIV for 14 years as well, and he had this cancer called myelodysplastic syndrome, which can turn into leukemia.
Oh, wow.
Yeah, so he got a stem cell transplant from his brother,
Okay.
and his brother had the double CCR5 mutation.
All right, so that's good news.
Yeah, it is, but there's a twist.
Oh.
The Oslo patient actually already had one copy of the CCR5 mutation himself.
Oh, wow. So he was kind of already partially protected?
Yeah, maybe, but we don't really know for sure how much protection one copy of the mutation gives you.
Huh.
But anyway, after the transplant, he got really sick with this thing called graft versus host disease, or GVHD.
Yeah, I've heard of that.
It's basically when the donor's immune cells attack the recipient's body.
Ouch.
Yeah, it can be pretty serious.
So what did they do?
They gave him immunosuppressant drugs to try to control the GVHD, and one of the drugs they gave him was ruxolitinib.
Okay.
Which is interesting because the Geneva patient also got ruxolitinib for his GVHD.
You don't think?
Yeah, maybe it's playing a role in these remissions, but we need more research to know for sure.
That's so fascinating.
Yeah, it is.
So back to the Oslo patient. Did the transplant work for his HIV?
It did. All of his immune cells were replaced with his brother's cells, and his viral load became undetectable and stayed that way. So 2 years after the transplant, he stopped taking HIV meds.
Okay.
And he's been off of them for 2 years now, and he's still undetectable.
2 years. Wow.
Yeah, it's looking really good for him.
That's amazing.
And they did a bunch of tests, and they couldn't find any signs of HIV really doing anything in his body.
So even though he might still have some tiny bits of HIV DNA hiding somewhere, it's not active.
Right. It's
Yeah.
not replicating, and it's not making him sick.
That's a functional cure right there.
It really seems like it.
So these two new cases are super exciting, but what are the big takeaways here? Like, what are we learning about HIV cure research?
Well, one thing that's becoming clear is that there's no one-size-fits-all solution. Like,
Yeah.
we've got these different cases with different CCR5 mutations, different conditioning therapies, different levels of GVHD, and we still don't know how much the size of the HIV reservoir matters.
Complicated.
It is, and one of the doctors who presented at CROI, Dr. Trayside, he said that we used to be really focused on CCR5,
Yeah.
but now we're starting to think more about how to actually get rid of the HIV reservoir itself.
Oh, interesting.
Yeah. And another doctor, Dr. Rubenstein, said that we need to look at all of these cases together and try to figure out what they have in common.
Right, to see what's really driving these cures.
Exactly.
Okay, so obviously this stem cell transplant thing isn't something we can do for everyone right now.
No, definitely not.
It's a pretty intense procedure.
Yeah, it's risky and it's expensive, and it's only really an option for people who have cancer already.
Right, you wouldn't go through all that if you didn't have to.
Exactly.
So what are scientists doing to find a cure that would work for everyone?
Well, one of the most promising areas is gene editing.
I've heard of that.
Yeah, so they're using tools like CRISPR to try to edit people's genes so that their cells don't have the CCR5 receptor anymore.
So it's like giving everyone that natural protection.
Yeah, exactly.
That's really cool.
Yeah, but there's one problem.
What's that?
One of the doctors at CROI, Dr. Henrich, he said that we can't just focus on CCR5
Why not?
because HIV is tricky.
Yeah.
It can find other ways to get into cells, so we need to target other parts of the HIV life cycle too.
Oh, I see.
Yeah, so that's something they're working on.
Okay, well, that makes sense.
And Dr. Henrich is actually doing some really interesting research with something called autologous stem cell transplants.
Oh, yeah, I read about that.
So in this case, they're not using donor cells at all. They're taking the patient's own stem cells and genetically modifying them in the lab to make them resistant to HIV.
So you're basically giving people their own cells back, but supercharged.
Exactly.
That's awesome.
Yeah, so they did a study with 11 people who had lymphoma and needed stem cell transplants anyway, and they found that the modified cells were able to survive and protect against HIV for at least a couple of years.
Wow.
And one of the patients even stopped taking HIV meds for a while, and their modified cells were able to keep the virus under control for 8 weeks.
Really?
Yeah, so it wasn't a complete cure, but it's really promising.
Yeah, for sure.
And the cool thing about autologous transplants is that there's no risk of rejection or GVHD.
Yeah, because it's your own cells.
Right.
So no need to find a donor.
Exactly.
That's pretty huge.
It is. It is, but we still need more research to see if this could work for everyone.
Okay, so the bottom line is that we're learning a lot from these stem cell transplants, both the ones with donor cells and the ones with the patient's own modified cells.
Right. It's all giving us clues about how to cure HIV.
Yeah, these two new cases,
Yeah.
the Chicago patient and the Oslo patient, they're really adding to the story.
Yeah, they are.
Like the Chicago patient showed us that you can still be cured even if the virus comes back at first.
Right.
And the Oslo patient is another example of how someone can stay in remission for years after stopping HIV meds.
Yeah.
But we have to remember that this kind of treatment is still only for people with cancer.
Yeah, it's not ready for prime time yet.
But it's giving us so much information to work with.
It really is.
So maybe someday soon we'll have a cure that works for everyone.
I hope so.
Yeah, me too.
It's an exciting time for HIV cure research.
It really is.
So for all of you listening, think about this.
Okay.
We've talked about all these different factors that might be important for a cure, like the CCR5 mutation, the size of the HIV reservoir, the immune response, even the cancer treatments. So what do you think is the most important thing we need to figure out to make an HIV cure a reality for everyone?
Yeah, that's a good question to think about.
Is it about targeting the virus directly, or is it about getting rid of the reservoir, or maybe it's about something else entirely? There's so much we still don't know, but it's an amazing time to be following this research.
That's for sure.
And who knows, maybe one of you listening will be the one to make the big breakthrough.
That would be amazing.
It would.
Well, thanks for breaking all this down for us.
You're welcome.
I learned a lot today.
Me too.
And I bet our listeners did too.
I hope so.
All right, everyone, until next time, stay curious.
And stay informed.
Bye!